Greetings to everyone. For those with an interest, our project is called Clinical Key for Electrical Stimulation in Physiotherapy and Rehabilitation. This project was founded by the Erasmus program of the European Union. Our project coordinator is represented by Pamukkale University from Denizli, Turkey, in collaboration with partners such as University of Craiova from Craiova, Romania, Hatay Mustafa Kemal University from Hatay, Turkey, Başkent University from Ankara, Turkey, Süleyman Demirel University from Isparta, Turkey, Šiauliai State University of Applied Sciences from Šiauliai, Lithuania, Burdur Mehmet Akif Ersoy University from Burdur, Turkey, and we also have collaboration with Turkish Physiotherapy Association from Turkey, Order of Physiotherapists in Romania, and Lithuanian Physiotherapy Association from Lithuania, and Estonian Association of Physiotherapists from Estonia. My name is Eva Ilie, and I am representing University of Craiova from Romania. My paper will be about healthy muscle physiology from Module 1, Healthy and Denervation Muscle Physiology. So an understanding of a healthy muscle physiology holds great importance for an extensive areas of activities, spanning from basic actions like walking, lifting objects, and to engaging in rigorous activities and exercises, and of course, excelling in athletic endeavors. So this chapter embarks in exploration, let's say so, of the fundamental principles underpinning robust muscle physiology, encompassing factors such as muscle contraction, energy metabolism, and the overall functionality of the muscle. So through this comprehensive examination, we aim to unreveal the intricate mechanism of the muscle, the way that it performs their intricate roles in supporting human movement and vitality. Each muscle has a specific functionality and of course possesses unique properties. So the anatomical structure of the muscle, including its shape, the orientation of the components fascicles, the spatial organization, as well as its connection with specific tendons, bones, aponeurosis, it's determined during the development since the beginning. So, being the final common pathway in Sherrington's model of control, motor control, the muscle stands as the ultimate executor of intricate network of neural processes. So the skeletal muscle has specific roles, such as maintaining the posture, assisting with involuntary actions such as breathing and reflexes, generates force and power during intentional movements, and produces a range of molecules called myokines that generates physiological impacts extending beyond muscle movement, portraying as well the skeletal muscle as an endocrine organ. So the skeletal muscle, accountable for facilitating voluntary movement, operates under conscious control, making it an integral component of our motor function. Comprised of muscle fibers capable of contracting and generating force, the skeletal muscle's functional prowess lies in its contractile properties. So its structural organization encompasses bundles of muscle fibers termed myofibers, and within each myofiber, multiple myofibril exists as the elemental cellular unit. These myofibrils are further subdivided into distinct sarcomeres. So they contribute to the muscle's remarkable contractile abilities. The skeletal muscles' intricate structure also include robust vascular and neural network. This is rich vascularization ensures efficient blood flow. So this is aiding basically in the delivery of oxygen and nutrients and the removal of waste products from the body. Simultaneously, the innervation by nerve fibers enables coordination signaling for muscle contraction and relaxation. And additionally, the skeletal muscle hosts also essential components for the metabolic and regulatory machinery. So this is required for energy production and cellular equilibrium. So this is reinforcing its role, the muscle role in maintaining overall physiological balance. All of these properties of the muscle, it's gaining in its dynamic capacity to generate force, to respond to neurological cues, to sustain metabolic dynamics and demands, and collectively enabling the diverse range of activities that our bodies are engaging in. Regarding the structural and functional components of the skeletal muscles, this can be segmented into three fundamental entities that propel the process of muscle contraction. So we have in the first place the neuromuscular junction, which is the connection between the nerve and the junction between the nerve and muscle. The machinery involved in excitation, contraction, coupling required to initiate the mechanical contraction. The sarcomere, that is the contractile apparatus responsible for force generation. If any of these important units can have a dysfunction or an impairment, this can lead to a muscle weakness and of course the loss of function. The structural arrangement of skeletal muscle is characterized by its constituent muscle fibers, also referred to as myofibers or muscle cells, along with their corresponding connective tissue. These myofibers are deliberately aligned in parallel to the line of force that the muscle generates. It's a configuration that contributes significantly to the muscle's overall strength and functional capabilities. Central to the muscle's contractile prowess is its internal myofilament architecture. So the intricate contractile properties are rooted in the organization of sarcomeres, the fundamental unit of this structure. Sarcomeres consist of interwound thick that are called myosin and thin that are called actin filaments, orchestrating the muscle's mechanical response to neurological signals or inputs. Remarkably, the sarcomeres are arranged and arrayed in repeating units along the length of the muscle, culminating in the distinctive striated appearance that skeletal muscle exhibits when observed through a microscope. This intricate arrangement not only imparts the muscle's unique visual pattern, but also underscores its remarkable capacity for controlled contraction, relaxation, to enable the varied spectrum of tasks that and activities that our body undertakes. Taking in consideration the capillary density that contributes to the differences in metabolic and endurance capacity between muscle fibers, we categorize the muscle fibers into three types. Type 1 are often referred to as slow twitched oxidative fibers and characterized by high oxidative capacity and resistance to fatigue. On the other hand, type 2 fibers are typically faster and powerful, but with lower oxidative capacity are prone to fatigue. Type 2A have an intermediate profile between type 1 and type 2X fibers. So we are talking now about type 2X or type 2B fibers that have the highest glycolytic capacity and are the fastest and most fatigable of all fibers. So the distribution of the fibers within the muscle is determined, of course, by genetics, by gender, age, and of course, training status. We will talk now about other important cellular components of the muscle fiber, such as the T-tubule system, that is a network of sarcolemma invagination within the muscle fibers that transmits the neuromuscular junction generated action potential for muscle contraction initiation. So it is also the one that ensures even distribution of the action potential through the muscle fibers. The other one is the sarcoplasmic reticulum, a specialized endoplasmic reticulum that stores and releases crucial calcium ions for muscle contraction. Muscle fibers mitochondria creates ATP, the adenosine triphosphate, that's the primary energy source for contraction. What we have here is a muscle fiber activation process image. It starts with stimulation of the muscle fiber, followed by an action potential that propagates along the two tubules, which triggers the release of calcium ions into the sarcoplasm. These calcium ions bind to the regulatory proteins troponin and tropomyosin that create an interaction between the actin and myosin filaments, which leads to the generation of the force. Of course, followed by the end of the stimulus. When this happens, the sarcoplasmic reticulum will reuptake the calcium ions, which would lead to the muscle fiber relaxation. Muscle fibers are primarily and mostly composed of proteins, including contractile ones, actin and myosin, regulatory ones, troponin and tropomyosin, and cytoskeletal ones, titin and nebulin, and forming about 80% of the fiber's weight. The remaining 8% consists of sarcoplasm housing organelles like mitochondria, the sarcoplasmic reticulum, and the T-tubule system, plus substances like glycogen and myoglobin, and the myofibrils from sarcomeres, the skeletal muscle's basic contractile unit, compressing the thin actin filaments and thick myosin filaments. Actin's lattice-like structure and myosin's globular heads create force and movements. Beyond the pivotal presence of actin and myosin, the composition of the sarcomere and sarcoplasm encompasses a multiple of additional proteins. These diverse proteins play an integral role in shaping the structure and orchestrating the function of skeletal muscle. Among the array of proteins, there exist those that are instrumental in various facets of muscle biology. The cytoskeleton, for instance, is fortified by specific proteins that bolster the structural integrity of the muscle fibers. The intricate process of excitation-contraction coupling, which underlies muscle contraction initiation, is facilitated by proteins that ensure seamless communication between nerve impulses and muscle action. Furthermore, proteins engage in energy release mechanism, and those involved in the generation of force and power further enhance the muscle's functional repertoire. So the interplay of these diverse proteins result in the harmonious functioning of the skeletal muscle. This intricate protein ensemble reflects the complexity of the skeletal muscle's operation. Regarding the muscle energy pathway, this depends mostly on slow twitch muscle fibers and fast twitch muscle fibers. The slow twitch muscle fibers are suitable for sustained aerobic exercises. The fast twitch muscle fibers are sustainable for bursts of anaerobic activity. The balance of the fast twitch muscle fibers and the slow twitch muscle fibers in the muscle determines its contractile properties and metabolic capacity. So we also have to take in consideration that capillary network supplies of oxygen to the muscle fibers, the myoglobin that facilitates oxygen to transport from capillaries to mitochondria and slow twitch fibers, and of course we have to take in consideration the physical training, the type of physical training that we are talking about. So for example, during a high intensity exercises, the body primarily will release on carbohydrates for fuel, while during low intensity, fat is the primary fuel source. We are talking about muscle force generation. So the orchestration of muscle movements extend beyond their anatomical structure and includes their functional role in generating the actual motion. The functional classification delineates various muscle actions based on their contribution to movement dynamics. So we are having agonist muscles, antagonist muscles, synergists, and stabilizers. The agonist muscles seize the spotlight as they contract to create the intended movement. Conversely, the antagonist muscles counteract the agonist action, imposing control or deceleration on the movement. Synergist muscles play a collaborative role, amplifying the movement's effectiveness by partnering with the agonists. So, on the other hand, stabilizer muscles pivot their focus toward joint or segment stabilization during movement, ensuring structural integrity. So some of the movements involve a combination of muscle actions, such as isometric contraction to stabilize the joint, while dynamic concentric or eccentric action occurs in other joint or body segment. Exercise training, especially when incorporating eccentric actions, can help reduce muscle damage. But can cause muscle damage if not executed properly. So, for example, for patients that go through rehabilitation for cardiopulmonary disease, eccentric actions can be beneficial because they require less muscle activation and energy consumption, and of course the oxygen consumption is less and compared to concentric actions that are given in a given level of. The utilization of adenosine triphosphate during muscle contraction is subject to a nuanced interplay of factors. These factors include the number of myosin heads engaged, the proportion of myosin heads forming cross bridges, and the rate constant governing the detachment of these cross bridges. This organization and orchestration determines the rate of ATP consumption and ultimately impacts in the efficiency of muscle contraction. Remarkably, in human muscle, the rate of ATP consumption exhibits variation across different types of fiber. So type 1 fibers characterized by their oxidative nature, exhibits the lowest ATP consumption rate. Following suit, type 2A fibers, known for their oxidative and glycolytic attributes, they are demonstrating an intermediate ATP consumption rate. In the other hand, in contrast, type 2X fibers, which predominantly rely on glycolysis, exhibit the highest rate of ATP consumption. A vital metric in assessing muscle function is the specific force. So a measure of the force produced by the muscle relative to its cross-sectional area. So the indicator offers valuable insights to muscle quality and its inherent ability to generate force considering its own size. Notably, the force generated through the interaction of actin-myosin cross bridges is not only conveyed along the length of the muscle, fiber, but also propagates laterally to neighboring fibers. So the force transmission mechanism, integral to muscle mechanism, serves a pivotal role in engaging movement, generating tension, and of course orchestrating the dynamic interplay between muscle fibers. The neuromuscular junction stands as a pivotal role in governing muscle contraction and preservation of muscle function. Its significant role relies in facilitating the transmission of signals from motor neurons to muscle fibers. However, disruptions within the neuromuscular junction have been linked to a spectrum of neuromuscular disorders, including conditions such as Lambert-Eaton myasthenia syndrome, myasthenia gravis, and amyotrophic lateral sclerosis. These disorders manifest as a consequence of compromised communication between motor neurons and muscle fibers at the neuromuscular junction. These disruptions translate in muscle weakness, fatigue, undermining the muscle's real capacity to create tasks in an efficient way. So given the pivotal role of neuromuscular junction in neuromuscular functionality and in association with various disorders, the exploitation and exploration of its structure and functional attributes emerges as a crucial need of investigation. This will, in depth, this in-depth comprehension of neuromuscular junction not only deepens our insight into the underlying mechanism of neuromuscular diseases, but also holds promise for developing effective treatments and therapeutic interventions. So, this is a way of understanding neuromuscular junction, and this will permit us to create new treatments and enhance our understanding of the intricate web of neuromuscular health and dysfunction. Regarding neuromuscular junction, the presynaptic region consists of Schwann cells, surrounds nerve terminal, and represents trophic support and acetylcholine release regulation. The nerve terminal, packed with synaptic vesicles, will be storing acetylcholine and activate zones for release. The postsynaptic region, mitochondria, Golgi apparatus, intermediate filaments, ion channels, voltage gate, sodium, represents the junctional folds with increased surface area that contains these cellular structures. The synaptic space represents a narrow gap between pre- and postsynaptic membranes, where the neurotransmitters are released from the presynaptic neurons and bind to the receptors on postsynaptic membranes, triggering muscle fibers. So, in synaptic basal lamina, which specialize in extracellular matrix, this lines the synaptic space and supports the neuromuscular junction. This will not be possible without their proteins components that are called laminin, collagen, and percolin. These are the references that I based my presentation on. This is our beautiful team, and I want to thank everyone for your attention.